Hello, welcome to the next A-level lesson from the conscientious biologist, Ben Gallagher. This one's membranes 1. It's quite tricky. There's a lot of information in here because we look at the biochemistry of lipids, and then we look at how lipids can form into bilayers, which are the main constituent part of membranes. This is the first time with me we've done biochemistry. So for those of you who don't like chemistry, don't panic. There's not too much of the theory of chemistry in it. You just have to memorize some of the structures, but I'm going to build those up in little steps for you to make it as straightforward as possible. As always, please do subscribe to my YouTube channel if you haven't done so already. And remember, you can follow me on both Facebook and Instagram. Thank you. So let's look at the basic building blocks that are going to start to come together to form the structures that then go on to form the structures of membranes. And there's two main component types. You've got glycerol and you've got fatty acids. Now, glycerol is relatively easy to memorize. It's 3 carbons in a row, and then you've got hydrogens above them, and you've got hydrogens to the sides of them. So they're pretty much kind of surrounded, but there's got to be something on the bottom as well. But it isn't hydrogens here. It's these OH groups or hydroxyl groups. Now you've got to have glycerol memorized along with most of the structures that we're going to look at now. So make sure you've got that. It's relatively straightforward. But to help you understand some of the biochemistry that goes with biology, what I've done here is I've looked at the 3 main elements that we use in this, which is carbon first, and carbon always has 4 bonds. So if you look up at the glycerol molecule, you can see that there's 3 carbons in the middle, all have bonds coming off the sides and above. They're bonded to 4 things. So if you're drawing something and you've put a carbon and you quickly count the bonds coming off it and you go, oh, I've only got 3, then you're missing something. Okay. Oxygen always has 2 bonds. So if you look at the oxygens up there, not as obvious with the oxygens in here, because the oxygens beneath, look, they've got 1 bond that goes up to the carbon, but the second bond is to that hydrogen. We don't show that bond because when we put an OH group, we always put the O and the H together. But technically, there should be a little bond line between the O and the H. So oxygens always have 2, and hydrogen only ever has 1 bond. Now, you don't need to understand the chemistry of that, so don't panic. Okay, let's move on to the fatty acid then. So at the top of the fatty acid, you've got this group. This group crops up a couple of times in biochemistry. You've got that carbon there in the middle. Coming from the side of the carbon, you've got a double bond to an oxygen. That's why there's those 2 lines. Double bonded to an oxygen. So tick, oxygen's got its 2 bonds. Above, you've got another hydroxyl or OH group. And the 4th bond from the carbon comes down. That's going to be joined to something else. But that specific group, a carbon, double bonded to an oxygen and bonded to an OH group, that's called a carboxylic acid group. Now, beneath the carboxylic acid group, you've got this, a long chain of hydrocarbons, meaning there's carbons with hydrogens on either side. Now, look all the way down, carbon joins to carbon, joins to carbon, joins to carbon, but each carbon, because it's got to have 4 bonds, has 2 hydrogens coming out the side of it as well, until you get to this bottom one, where because that carbon isn't joined down to a carbon, you just close off the end with another hydrogen. Hydrogen that only requires 1 bond, so that just fits on nicely. So that's a fatty acid chain. Now, there's 2 key kind of types or variants with fatty acids. You can have this one, which is called a saturated fatty acid. The word saturated kind of means full of. So that fatty acid there is full of hydrogen. It's got as much hydrogen joined onto the carbons as it possibly can. What we're going to do next to it is we're going to do an unsaturated fatty acid by putting exactly the same molecule next to it. But watch what I'm going to do between a couple of the lower carbons. I'm going to remove the single bond and I'm going to put a double bond in there. Now, because there's a double bond between those 2 carbons, now count the number of bonds coming off those 2 carbons. Each one of those 2 attached to the double bond have now got 5 bonds. 2 in the double bond, a hydrogen on one side, a hydrogen on the other side, and a carbon to the carbon either above or below it. So that can't be. So how can we get this to work? Well, each of them will lose a hydrogen. So we've lost hydrogens on this one. So it's no longer saturated with hydrogens. If we broke that double bond, we could put some more hydrogens on there. Now, we could also put more double bonds in between the carbons. And the more double bonds that go in, the more hydrogens you'd have to take away. So we're making it more and more unsaturated. Again, just meaning it's unsaturated if you could put more hydrogens on there, until you got to a point where you came to the one closer to me where it's totally full, can't take any more. That's a saturated fatty acid. Now, when we get to membrane structure in the next lesson, we'll talk a little bit more about the differences between saturated and unsaturated and why they're there. But for the time being, you just need to know these three different structures. You must know the glycerol head, and you must know a saturated fatty acid, and that if you put in some double bonds between the carbons, you lose hydrogen. So unsaturated ones contain the double bonds. Okay, take a screenshot of this because it's a really important start point. Right, we're now going to start to put those two things, the glycerol and the fatty acids, together to make the main type of lipid molecule. Lipids, you might remember from GCSE, are fats, and membranes are actually made of lipids. So membranes are basically made of fat, okay? And the main one is this triglyceride molecule. So what I'm going to do, I'm going to pull up the glycerol molecule again up there, and beneath it I'm going to put one of the saturated fatty acid chains. It doesn't matter that it's saturated or unsaturated, I've just randomly decided to pick saturated because it's the easiest one to draw. Okay, now what I want you to do is look at what's going to happen inside that yellow circle there, okay, because that's where the business is going to take place. We need to bond the glycerol molecule to the fatty acid. So watch where that circle was. What we're going to do, we're going to take away the two hydrogens from the adjacent OH groups. We're going to take away one of the oxygens. So think what we've taken away. Two hydrogens, one oxygen, H2O. Well, H2O, you should know, is water. So when these two react together, they give off water in what is called a condensation reaction. Because condensation is water formation. When you breathe against a cold window, you see condensation forming because it's water vapor in your breath forming on the window. Okay, so it's a condensation reaction because we've lost H2O. And then lastly, all that happens to make all the bond numbers work, the carbon of the glycerol bonds directly to that oxygen that was in the OH group of the carboxylic acid group. So by losing that H2O, we freed up space to bond the carbon and the oxygen. So we've created a bond there. That is called an ester bond, okay? And an ester bond that formed by a condensation reaction. Now, in a glycerol molecule, you have three of these ester bonds because you have three fatty acids that join on there. So if we put them on there on the diagram, look, I'm going to do exactly the same, remove H2O from each one of the groups and put on a new fatty acid. Now it starts to look a bit muddled and confused at the bottom because my diagrams have overlapped a bit, but you can see what's happened here. We've just got three fatty acids joined by three ester bonds to the glycerol molecule. So you've got one glycerol head and you've got three fatty acid chains coming down from it. Now remember, each one of those ester bonds formed by a condensation reaction, so each one of them will have given off a water molecule. So what you actually get, you'll see I've changed on the side, there is three H2O molecules given off in the creation of a triglyceride. Or to flip that around the other way, because triglyceride are fats really, so if you eat too much bad food and you get fat, it's these things, these triglycerides, that you're storing in your fat cells. So what we could say, flip it around, is that it takes three water molecules to break them down. If you put a water molecule back into the ester bond, replenish the hydrogens, replenish the oxygen, they can break that bond. So it's very important if you're trying to break down fats to make sure you're drinking plenty of water, get hydrated, because you can't break down triglycerides without adding the water that initially was given off during the condensation reactions. So take a screenshot of this or re-watch it if you need to see again the details, because in the exam you're likely to be asked to draw an ester bond or fill in the blanks if they give you some of that diagram, or just describe what happens in an ester bond. So make sure you know this. Take a screenshot if you need to. Now the specific lipid molecule that's present in membranes is called a phospholipid, and you can get a phospholipid from a triglyceride. So we're going to build up a phospholipid now. Now you don't need to know the full biochemical structure like this for a phospholipid. You do need to know it for a triglyceride. You could be asked to draw one. Very unlikely that you'd be asked to draw one in full because it's a massive molecule. It would take you ages, but you might be asked to fill in the blanks or describe what's missing or whatever. But you don't need the full structure for a phospholipid. What we're going to do for a phospholipid, we remove one of the fatty acid groups and we replace it with a phosphate group, which looks like that biochemically, but you don't need to know that, remember. All you need to know is that phospholipid is basically a triglyceride where one of the fatty acid chains has been replaced with a phosphate group. Now, if you're drawing that at a more simplistic way, we just draw it like this. We put the glycerol head as kind of a blob, and we put the two fatty acid chains that come off from it. When we're drawing phospholipids, because you've got to draw lots of them if you're going to draw a membrane, we generally don't bother to show the phosphate group, and we certainly don't draw the full biochemical model that you can see over there. So this little blue diagram next to me, you're going to see this popping up a lot in the rest of this presentation and the next video lesson on transport through membranes. Again, take a screenshot of this, but remember you do not need to memorize that structure over on the far side. So let's move on to formation of membranes. And to understand that, you've got to understand how lipids behave when they're in water. So if I pull my phospholipid diagram back up there, and we've got the glycerol head and we've got the fatty acid chain, same as I labeled on the last one. But what we're going to look at now is the properties of those. Now, the glycerol head is said to be water loving, or the correct word for that is hydrophilic. Philia from the Greek, meaning love. So hydrophilic glycerol head. I would suggest when you're labeling lipids, maybe label it as hydrophilic glycerol head. Then you're ticking all the boxes for the examiner and you're definitely going to get the marks. Now, the reason why the glycerol head is hydrophilic or water loving is because water is also, kind of glycerol loving because they're both polar. So as I've written over here, glycerol and water are both polar molecules. Polar means they've got positive and negative parts. They're like North Pole and South Pole. They've got opposite ends. Now, if you've got opposites on you as a molecule, you can be attracted to other molecules. So on a water molecule, there's a positive and a negative side. On a glycerol molecule, there's a positive and a negative side. So the positive side of the water and the negative side of the glycerol can be attracted to each other because opposites attract. So basically, when we say that the glycerol head is hydrophilic, it's simply because it's polar, just like water, so they can attract to each other. Now, the fatty acid chains are not polar, so they're called hydrophobic or water hating. Now, that's really important when you consider what they do in water. So if I put a big container of water here, let's say I've got a glass of water, I filled it right up to the top. If I had a load of lipids massively upscaled so they were visible, and I chuck those lipids into this glass of water, there's only a few things they can do. The logical, obvious thing is this: to protect the fatty acid chains, or because the fatty acid chains hate the water, they don't want to be anywhere near it, lipids might float on the surface like this, with their heads in the water because they love it, and the tails out of the water because they hate it. We said at the start, lipids are fats. This is why fats float on water. If you took some cooking oil from your kitchen, which is full of lipids because it's fats, you poured it into some water, it would sort of fall into the water and then all bubble back up to the surface and form a layer on the surface. If you could see that really, really clearly, what you'd be looking at on the surface is the little fatty acid chains poking out above. So this is why fats float, because of the properties of the phospholipids. Now, you might also note, if you put oil in water, let's say you're washing up some greasy saucepans or something, you might notice that when you pour the water out of the sink, that the sink feels greasy all around the sides. That's because another thing that lipids can do is they can use the container itself, like this, to kind of form, to protect the fatty acids by forming a layer using the sides of the glass. That's why if you're washing up, you feel down to the bottom of the washing up pole, you can feel it's greasy on the bottom, because that's the lipids kind of forcing themselves against the side, trying to get their fatty acid chains behind them protected. And you can see where I put it white. No water can get in there. It repels all the water out. So technically behind the glycerol heads, you've kind of got a little dry bubble behind. Now, it's this third thing I'm going to show you that's really important for membrane formation. If the lipids don't stay on the surface or push themselves against the sides or the bottom, what they can do is float in the middle, as long as they all work together as a team to form a layer of heads around the outside to keep their fatty acid chains on the inside. Now remember, you're looking at a cross section here. Try and think of this in 3 dimensions, that blob in the middle there is actually a sphere, a little ball. And on the outside of the ball are all the glycerol heads, so that trapped inside would be the fatty acid chains protected. It'd be a tiny little droplet, which is exactly why you see tiny little droplets of fat in water. If you've got any fat in your water, swish it all up with your hands, look at it, you'll see millions of tiny little droplets. Now those droplets will quickly blob to each other and form bigger droplets and get to the surface and float. But these tiny droplets of fat are called mycels. So that is a mycel on there. This is really important for what we come on to next. So take a shot of this because it's so essential for membrane formation. Now let's take that same mycel that we've just looked at, but what I want you to imagine now is instead of that sphere like that, imagine if we could extend the sphere out like that, still just protecting all the fatty acids on the inside, but if we could extend it out and extend it out in all directions, again, try and think of this in 3 dimensions. So imagine if this diagram kind of extends back into the screen and out towards you, so you've got a big kind of sheet of protected lipids all together now. Now, if we take the ends off this, I can label that because that is a phospholipid bilayer. It's 2, that's the bi, layers of phospholipids. Now, phospholipid bilayers are the essential integral part of membranes because if you look at it, you can have water outside on one side of that, and you can have water on the inside. In other words, you've made a skin that can hold water out but still contain liquid within, such as cytoplasm, to make a cell. If I show you what I mean here, if I shrink that right down, so that piece of phospholipid bilayer is now there, if I just extend that right round, this blue line now is representing cell membrane with cytoplasm inside that contains water. Outside of the cells, so here kind of next to me, could be the tissue fluid that's inside your body. You've essentially made a watertight layer that can create a compartment that is your cell. That's the basics of membrane structure. So make sure you've understood that, okay? Take a screenshot of this one if you need to, but make sure you understand how the phospholipid bilayer basically evolved from a mycel stretching out into sheets. There's a lot to memorize there, especially with those biochemical structures of the glycerol, the fatty acids, the triglyceride, and the phospholipids. So go back, watch through it again. Please take some time to draw them out, test yourself, check it against your screenshots you've taken, see if you've got the ester bond right. Loads of people, loads of students over the years that I've had always forget to put the double bonded oxygen in the ester bond on the carbon of the carboxylic acid group. Don't forget that. But just practice and practice and practice until you can draw them out from memory. From here, go on to membranes 2, which is a direct follow-on. We look at how you can transport things through membranes. If you like this video, please do give it a thumbs up, subscribe to my channel if you haven't done so already, and do think about following me on Facebook and Instagram for lots of additional biology to round out your knowledge. Thank you.